Okay, welcome back to the second part of the series, that is the structure of RNA. Till now we have been discussing about what is the structure of DNA, what are its properties of DNA, and also we have discussed the replication property. Okay, three part we have discussed, that is from what are the enzymes, then what is prokaryotic DNA replication, eukaryotic DNA replication, and telomerase replication. With this we came to an end with the DNA series. Now we are going to start with the RNA. Then after the structure of RNA, we will be discussing about the transcription, from how from the DNA RNA is synthesized, and finally that is translation, from how the from RNA proteins are synthesized. With this we will be ending a series of the molecular biology. So let's start. First, before understanding the transcription and translation process, it is very important to understand the structure of RNA, because without knowing the structure of RNA, you will not be able to understand how the synthesis process is going on. So let's, in this part of the video, we will be only considering the structure of RNA, and in the next part we will be discussing about the transcription process. So let's begin. As you all know that the RNA is a nucleic acid, because in the first part itself, in the DNA structure only, I have discussed that the RNA is a single stranded structure. Okay, and it is also a nucleic acid, and it is known as the ribonucleic acid. Why it is called ribonucleic acid? Because it contains, that is, it is single stranded. And why it is called ribonucleic acid? Because it have ribose as a sugar molecule. This is the important point. Okay, and it is folded upon itself entirely or in some region. You can see this is the single stranded structure of RNA, whereas we have discussed DNA, which is a double stranded structure. Okay, but also there is an exception here also. In some viruses the RNA is double stranded. In which viruses? Like the reovirus and wound tumor virus, the structure of the RNA is mainly double stranded. Okay, and DNA was only single type. Different forms of DNA was found, but RNA are of three different types. Cellular RNA are of three different types. First is the messenger RNA, ribosomal RNA, and third is the transfer RNA. Why the name is called so, we will be discussing each one in detail. Let's begin with the difference, or before that let's first discuss about the difference between DNA and RNA, so you will be very much clear that what is the DNA and what is RNA. Okay, let's begin. First is that most of the genetic material is the, usually we see DNA as a genetic material, but in some virus the RNA also function as a genetic material. This is an important point. And DNA is double stranded, okay, whereas the RNA is single stranded, exception in case of the virus. Okay, sorry. Next is the pentose sugar. As we have already discussed that the DNA is deoxyribose sugar, okay, and pentose sugar, that is RNA, is ribose sugar. Okay, I have already shown the structure in the DNA part. You will see that part and you will understand. Okay, next that is important is the base sequence. If you remember that the DNA had 4 nitrogen bases sequence, that is adenine, guanine, cytosine and thymine. Here also 4 nitrogen base sequence will be present, but instead of thymine, uracil will be present. This is very, very important. All the rest, that is adenine, guanine, cytosine, will be same, except thymine, that is uracil, will be present. Okay, and the base pairing as such, adenine was in DNA was pairing with thymine, but here adenine will be pairing with uracil, okay, and guanine with cytosine as usual. Next, coming to certain other base differences, like the base, there are certain more uncommon base sequence will be found in case of RNA, but in case of DNA there are not such much. And DNA, as I have told you in the beginning, that DNA is only of one type, but RNA is of three different types, that is ribosomal, messenger and transfer RNA. And most of the DNA are found in the chromosome, Messenger RNA is only found in the chromosome and found in the nucleus, whereas rRNA and tRNA are also found in the chromosome and found in the cytoplasm. Okay, this is not much difference. Next, coming to the point, that is denaturation is possible in case of DNA, but practically a complete denaturation of RNA is not possible. These are certain major difference between DNA and RNA, and there are certain other also point, like if you want to refer, you can pause the video and note down all these differences. I have discussed the major differences. Now moving to the first part of the RNA, that is ribosomal RNA. Why ribosomal RNA is called so? Because it is found in the ribosome. Ribosome is a part of a cell. Okay, when we will be discussing cell biology, we will be discussing about ribosome, but ribosome is a part of a cell which is the center of protein synthesis. Why I am saying so? Because here the protein synthesis takes place. When we will be discussing about the translation, you will be better clear with the idea. And one more important thing is that the ribosomal RNA forms 80% of the total RNA. That is, the RNA is of three types: ribosomal, messenger and transfer. Out of all these three, the major is the ribosomal RNA. And ribosomal RNA bases are complementary to that of the DNA sequence, okay, from where it is synthesized. So DNA is the main synthesis site from where the rRNA will be formed, okay, so that the base sequence are matching. And in prokaryote, rRNA are formed from the DNA, and eukaryotes, the rRNA is formed from the nucleolus. Okay, this is the difference. Next, coming that the rRNA is single stranded. As you know, RNA is a single stranded, so rRNA obviously single stranded, and they, in some part they will be coiled upon itself and some region will be uncoiled region, okay, in case of the ribosomal RNA. And the as you know, the ribosome consists of two units. One is the proteins, another one is the RNA, okay. So 70S ribosome, there are two types of ribosome. One is 70S, which is found in case of prokaryotes. Second is 80S ribosome, which is found in case of the eukaryote. This is important to be noted down. Again, the 70S ribosome in prokaryote is divided into two subunits, that is 30S and 50S. All this point you need to be very clear and very thorough with it, because this part we will be discussing about in the translation. What is 70S, what is 30S, what is 50S? If you don't know that, it will be creating problem for you to understand the process of translation. Okay, this is not very difficult to understand. 70S is a ribosome which is found in case of prokaryote. It is of two subunit, that is smaller subunit, that is 30S and 50S. What is S is the sedimentation coefficient. Okay, and if you add them, they will not form the 70S. So don't remember like that. Okay, 70S is of two subunit, and each subunit has the rRNA. That is, 30S subunit has 16S rRNA, you can see here, and 50S have two rRNA, that is 23S and 5S rRNA. Okay, this is very important to be noted down. Next, coming to the 80S ribosome, which is found in case of eukaryote, has two subunits, that is 40S and 60S. Okay, 40S have 18S rRNA and 16S has 3 rRNA, that is 28S, 5.8S and 5S rRNA. So this part is very important for you to understand. Okay. Next, moving to the second type of RNA, that is messenger RNA. Why it is called messenger? Because it is carrying, what is the role of a messenger? To carry information or to send certain information. Okay, you will be better knowing with that. If you send someone message, what you are sending? Some information or certain things. Same role is performed by the messenger RNA. What information it is carrying out? So the material or the information which is needed for the protein synthesis is carried by the messenger RNA. Where does it carry? It carry to the ribosome, because in the starting only I have told you that ribosome is the site of protein synthesis. Okay. So from DNA, which is the storehouse of information, the messenger RNA will carry the information from DNA to the ribosome. This is a very interesting phenomenon which is performed, and it is important function performed by the messenger RNA. Messenger RNA term was given by two scientists, that is Jacob and Monod. Okay, Jacob and Monod, and it only formed the 3 to 5% of the total RNA, and it is also single stranded, and bases are also present, but the base pairing does not take place. This is an interesting feature to be noted down. Why? It will destroy its biological activity. Then you will be wondering that base pairing if does not takes place, then how will be stability is maintained. So messenger RNA forms the base pairing with that of the transfer RNA or with that of the DNA through which it is synthesized. Okay, so for that purpose only the base pairing takes place. Next, the gene transcribes its own mRNA, which is very important. If one gene, this point is very confusing for many students, but if you understand that if one gene codes for one mRNA, it is called as the monocystonic. Okay. If many genes code for one mRNA, it is called polycystonic. In one way you may understand. One gene, one mRNA, monocystonic. Many genes, one mRNA, it is called polycystonic. Next, there are certain structures which are very much important for the messenger RNA. What are the structures? First is the cap. You can see here. Okay, wait. This is the cap. Okay, first part is the cap. Second is the non-coding region. This first one is the cap region. Okay, second is the non-coding region 1. Then is the third is the initiation codon. Then is the fourth is the coding region. Then comes the non-coding region, fifth one. Okay, sixth is the termination codon, and seventh is the poly-A sequence. So what are these? These are the part of the mRNA. So when we will be discussing about the transcription process, we will be discussing in detail that why this cap is needed and why this poly-A sequence is needed, because from the DNA when the mRNA is formed, it is known as pre-mRNA. All the structures are not present, like the cap and the poly-A sequence are not present. So to form a complete mRNA or to form a full mRNA, stable mRNA or mature mRNA, that is capping as well as poly-A sequencing is needed. We will be discussing this when we will be discussing about the transcription process. Before that, why the capping is needed? Because the cap is added in the 5' end region. Why? So that capping determines how well the ribosomes will get attached to that of the mRNA structure, because at the end of the the mRNA need to be carrying the information to the ribosome. The ribosome needs to be attached to the mRNA so that the protein is to be made. So for that purpose, the cap is added at the 5' end. You can see this is the 5' end, this is the 3' end. Okay, this is the 3' end, this is the 5' end. So to the 5' end region, basically the cap is added so that the proper binding of the ribosome takes place. Then next comes the non-coding region 1. It is mainly rich in the adenine and uracil region, and why it is and it is made up of 10 to 100 nucleotide. Why it is called non-coding? Because it does not take part in the process of protein synthesis. As next is the initiation codon, that is AUG. What is the role of the initiation codon? From here the actual protein synthesis will begin. Okay, the ribosome will no doubt bind in the cap region, but the actual transcription process will start from the initiation codon, that is AUG. Next is this is the coding region. This blackened end is the coding region where which will take part in the protein synthesis process. Next comes to the second non-coding region, that is 50 to 150 nucleotide. It contains adenine, uracil and adenine sequence, that is AA-UAA sequence. Next coming to the termination codon. So if the synthesis of mRNA needs to be takes place, there should be a certain end to that, otherwise the synthesis will be continually going on. So for the end of that, there need to be a codon which take part, that is UAA, UAG or UGA. That is the 3 codons are present which takes part in the termination, which we will be discussing in detail in the transcription. Next comes to the poly-A sequence. The poly-A sequence is then attached to the 3' end, that is 20 to 200 nucleotide, and this is added before the mRNA leaves the ribosome, okay, before the mRNA reaches the cytoplasm, this poly-A sequence is added. So now the mRNA is prepared. Okay, now the mature mRNA is prepared. So this is the structure of the mRNA. Next, coming to the last part of the RNA, that is transfer RNA. This is the second most common RNA. First most common is the ribosomal RNA, second comes the transfer, and the least is the messenger RNA. And what is the role of transfer RNA? As the name suggests, it transfers the amino acid, because we all know the amino acid together forms the protein. So the basic subunit is required so that the protein will be formed. So these amino acid is carried by the transfer RNA to that of the, where it is carried? It will carry to that of the messenger RNA, so that the messenger RNA will synthesize the protein. Okay, and it is to be noted down that each amino acid is carried by a specific tRNA. Specific tRNA will carry each amino acid. So there are 20 tRNA for 20 types of amino acid. Okay. In the secondary structure of the tRNA, this is the secondary structure of the tRNA, have 5 arms. You know that the tRNA is single stranded. So due to single stranded, they are forms a loop like structure. They are folded upon each other. As a result, the 5 prime and the 3 prime end of the strand comes close to each other, and they form 5 arms in which certain stem and loops are present. In certain arm only stems will be present and no loop. In certain case only loop will be present, no stem will be present. So each one of them we will be discussing one by one. So first we will be discussing about the acceptor stem. What is the role of the acceptor stem? So acceptor stem is the this arm, this stem. Okay, it has only stem but does not have any loop. Okay, and you can see here in the 3 prime end there is a sequence called CCA sequence. So in this sequence the amino acid will come and bind it. That's why it is called acceptor stem. It will accept the amino acid in this sequence, that is CCA sequence. CCA, like this it will be present. Okay, because we read the sequence in the 5 prime to 3 prime direction, that's why we will read like this, not ACC. CCA sequence. Okay, next comes the D loop. Why it is called D loop? Because it has dihydrouridine. Okay, dihydrouridine, and it consists of mainly, that is D loop, and it has many nucleotide and guanine residue is present. Okay, it is also called as the DHU loop. Okay, it has both stem as well as loop, and it has 7 to 11 unpaired bases will be present. Next comes the variable loop. It is varies. Why it is called variable loop? In some secondary structure you will be seeing that only stem will be present like this. Here only loop is present, but in some cases only stem will be present, no loop will be present. But in some secondary structure only loop will be present, no stem will be present. That's why it is called variable loop. Next comes the anticodon loop, which is very important. It contains the base sequence which will be complementary to that of the mRNA sequence. Okay, this is we will discussing when we'll be reading about the protein synthesis. Next comes the T loop, or you can say T psi C loop. Okay, T psi C loop, and this loop is very much important in the recognition of the process. Okay, for the recognition of the ribosome, this is very important. So T psi C loop is important for, why it is important? For the recognition of the ribosome. So this is all about the structure of the tRNA. Okay, this is all about the structure of the RNA. Okay, different types of RNA. Hope you are clear with the structure of RNA, because in the next part we will be discussing about transcription. So with that we will be discussing each one, that is what is transcription and all the process. Before that you need to be clear about the structure of RNA. So that's all for today, and please do subscribe to my channel, share as much as you can among your friends, because these are very basic, asked in exams as well as important for your entrance or MSc or BSc level. These are very basics. Okay, and like, if you like, and share and subscribe, and also comment if you have any suggestion regarding, or if you doubt regarding this video. And that's all for today. Thank you and have a nice day.